talks. And for now, I'm going to invite Dr. Eline Chauvet-Piat, and she will be talking on CTNNB1 natural history and AADC gene therapy experience, a contribution from France. The floor is yours.
Good afternoon, everybody. Thank you very much for letting me speak today. Thank you especially to Spella. Is it going on? Yeah. I'll wait a few seconds then. Yeah, thank you. So I'm Dr. Eline Chauvet-Piat. I am French from origin, but I'm working in Switzerland, Geneva. And currently, I'm doing a fellowship in movement disorder in Montpellier in France with Professor Roberti, which was invited as well, but had another commitment. We prepared this talk together. There's 2 points that Spella wanted us to expose. The first one is our CTNNB1 experience in France, because we have quite a lot of patients, and also our AADC gene therapy experience. So this is the paper that we published at the beginning of this year about CTNNB1 natural history in France. It was a retrospective study on patients with monoallelic pathogenic or likely pathogenic variants. We had around 25 patients. I thought it was the only patients we had in France, but I've met more now in France as well, and there's more coming in Montpellier to get inside this natural history. They came from multiple French centers in France, and the mean age was 10.9 years. Data collection were with medical records and a structured questionnaire that we sent to them to be filled in with their neuropediatrician in their own city. And we did clinical evaluations and reviewed their brain MRIs with 2 of our neuropediatric radiologists. I'm not going to go through all of the results, but maybe the main ones. So talking of neurological evaluation, we determined it was a complex motor phenotype with 3 main points. The first one is gait disturbance, which is a mix of unsteadiness, hypertonia of lower limbs. May it be dystonic or non-dystonic. There's more spasticity in some, more dystonia in some, and also this dystonia of other body parts that can also participate in gait disturbance. The second one is exaggerated startle reflex, and some of our patients have the fear of falling, and that also contributes to their gait disturbance. And the final one is that it may also evolve over time, and we need more adult patients to understand exactly where it's going. On the ophthalmologic point of view, so you know the importance of fluorescein angiography, but we wanted to make that point as well. So as you know, early stages may be asymptomatic, only detectable by a fluorescein angiography, and the treatment has to be done as soon as possible with laser photocoagulation. And if you see in our cohort, only 4 patients were diagnosed, but 3 of 5 with angiography. So we feel like fluorescein angiography, if it's not done, we're not going to be able to diagnose them and treat them. And the third point is about radiology. So we expanded the CTNNB1 brain MRI non-specific findings with a thickening of the anterior commissure present in 42% of our patients, frontal lobe hypoplasia in 47.7% of patients, and the widening of the superior vermin sulci in around half of our patients. So now moving on, we're also conducting a CTNNB1 pilot study with levodopa. So levodopa is not a brand new thing. We use that a lot in neurology, but there are no that much studies about it in CTNNB1 patients. There has been a cohort of Canadian patients, 5 Canadian patients that were tried with that. But Professor Roberto, way before I actually came into that, was already writing a protocol, and then we heard about the gene therapy, and we said, it's okay, we can still go along with that, and maybe some are responders, and we want to know exactly who. Is it some certain variants, or is it something else? But we're doing it right now. So it started in April. It's a single-arm proof-of-concept trial, monocentric, only in Montpellier. It's during a whole year, and it's obviously off-label use of L-dopa and carbidopa. We had treated before 2 of our patients in Montpellier off-label, and they actually quite had good evolution on the motor side, so we feel it was interesting to try and see a bit more. So it's children aged between 1 and 15 with obviously a pathogenic variant of CTNNB1 and with presence of dystonia, as it is the most known effective result with L-DOPA. So primary objective is to assess the effect of L-DOPA on motor function using the GMFM-88, and we have secondary objective with cognitive development, quality of life, patients and caregivers perspective, global clinical benefit, and treatment safety and tolerance. So how it goes, we give L-DOPA and carbidopa with a gradually introduced over 3 weeks. The target dose, it's 3 milligrams per kilo per day. Some will use more, I'm sure, but that's how it was written. And then we evaluate them at D1 and then 6 months and then 12 months. And so in the meantime, while the CTNNB1 knowledge is growing everywhere, we also have an AADC experience, and we feel like it's preparing the grounds for future therapies as well. So AADC disease is an enzyme deficiency disease that causes some dopamine, serotonin, and epinephrine to not be as sufficient in the brain. And phenotype are quite severe. So there's severe early motor impairment with hypotonia, dystonia, and ocular gyrate crisis, autonomic dysfunction, and also global neurodevelopment delay. It's an ultra-rare disease. We have around 10 patients in France. Treatments are very limited, and as it is a monogenic disease, it's an ideal target for gene therapy. So how we do gene therapy, we deliver a functional DDC gene directly into deep brain structures to restore dopamine production. We put it in the putamen. Some will use the substantia nigra, which is a bit different from CTNNB1 gene therapy, which is intraventricular. And we use a vector, which is AAV2. AAV vectors, as you know, are non-pathogenic, highly stable. They do not initiate an immunologic response. They stay inside the target tissue, and also they have a long-term expression, which is what we need. So what's the technique? It's the stereotactic neurosurgery with intracerebral injection into putamens. We put 2 per putamens. It's MRI-guided, and we infuse 4 times 80 microliters. So international data had done that before us, and Taiwan was the first, then Japan and the US, with quite good results on motor function and also on awakening of the patients and also reduction of ocular gyrate crisis. And these are some of the results, the AIMS score, the Bailey 3 language score, and also the ocular gyrate reduction of crisis. So after we had the international proof of concept data, we started building a team of experts in Montpellier in France in February 2020, and then in December 2020 we had the first European patient treated in Montpellier, and now there's 10 patients treated and 2 pending, 1 from Portugal and 1 from Greece, which we are getting in our hospital next week. So how it goes, the clinical pathway, we ask for clinical evaluation beforehand, and then we have a national expert discussion with also the family and the caregivers and the neuropediatrician of the child. Then we admit them 10 days before surgery, and then they go in ICU for 24-48 hours, and they are discharged from hospital around 10 to 15 days after. So what's our patient profile? This is our first 8 patients. They had real severe phenotype with no head control, minimal voluntary movements, frequent ocular gyrate crisis, and sleep disturbances. And we saw quite a modification of the disease trajectory. So about movement improvements, they acquired head control. They would be more alert and have a better sleep, and also ocular gyrate crisis would have a significant reduction. What about safety? Our main adverse effect was dyskinesia that started around 15 days after the surgery and could last weeks to 10 months. So as I said, they're discharged from hospital at around 10 to 15 days. So it's after we say goodbye to them. So it's very important that we keep in touch with the neuropediatrician in their own hospital, and that we continue knowing how the child is going. And this is maybe the thing that we share with CTNNB1 patients. If you're going to have patients from everywhere in the world, we have to keep in touch because they're going to have to treat all these side effects afterwards. So yeah. And that's that. So this is our CTNNB1 experience and our AADC gene therapy experience. It's only to share where we hope that this is just to enhance CTNNB1 expertise. And we also know that this is an international collaboration and we cannot do it on our own. Thank you very much.